So let's get into a little bit of when we talk about disease. There's a lot of people who just say, "Hey, we get older." Of course, that happens to everyone. I think that the conversation is changing a little bit to this idea of like aging is a disease. But I want to break down like what exactly is happening as you age, and kind
hmm
of why there's a growing portion of people who believe that we can actually treat that, prevent it, delay it, kind of all these different aspects. And so first, let's maybe start with like the 9 hallmarks of aging. I know that you guys have done a ton of work of identifying these and explaining them and kind of elaborating on it. But what does it mean when you talk about the 9 hallmarks of aging?
Yeah. So I think this is really the right place to start. So the 9 hallmarks of aging are basically 9 different types of molecular damage that build up in our bodies as we get older. And so, you don't just get cancer, right? The moment, it doesn't just appear. It happens over a very long period of time where different aspects of our bodies are breaking down little by little. In the case of cancer, you have DNA mutations that accumulate one after the other, creating an evolutionarily selective pressure for some cells to outcompete others until you kind of, run this cycle long enough that you have this uncontrolled tumor. But the damage that starts all of that are mutations in our DNA that happen randomly, even in our 20s and 30s, that are constantly happening way before we get cancer. And there are 9 of these types of damage that include, chronic inflammation that happens, just more and more as we get older. Buildup of aggregates, like the aggregates that cause Alzheimer's disease. But there are more than just amyloid and tau aggregates that are building up all the time. And the machinery that we have, the recycling mechanisms for chewing up those aggregates, degrades over time as our cells and our tissues get older. And so there are this group of scientists in this emerging field called geroscience, right? The study of what makes us age, have mapped out way more than 9 particular mechanisms, but they fall into these 9 categories that we call the hallmarks of aging, which define the different types of damage. And what we've been able to do with those 9 categories, which were only kind of distilled into a single thesis about 7 years ago to 2013, 2014, what we've been able to do since then is you pick one of those types of damage and you tweak it, right? Either preventing it from building up or removing it from an already aged animal. And we see this amazing thing happen, which is you can take a mouse, for example, tweak one of these hallmarks of aging, and it just lives longer and healthier than it ever would in the past. So instead of living, for a mouse like 2.5 years, it's living 3 or more years instead. And instead of getting sick at 2 years so that it dies at 2.5 years, it's only getting sick at, 2.5 or close to 3 years. And that change is fundamentally what gets me excited about this space, because that would mean, if we could translate that into human biology, like it would be way better than like a magical cure for cancer or something like that, as far as like the number of extra healthy years of life we could distribute to the population. We can talk about that a little bit more in a second.
Yeah. So what I want to talk about is just, at a very high level, when you talk about molecular damage, just explain that very quickly for people, because I think that that's somewhat table stakes in the kind of the scientific community and the longevity community, but there's a lot of people who won't understand what that actually means.
Sure. So, we're all just collections of cells. Cells are bags of fat containing water, protein, DNA, and other molecules, right? And the molecules that make up our bodies are changing dynamically all the time. And so we're constantly being assaulted with, UV rays from the sun, or crap that we put into our bodies that we eat, or even just like random changes as we get older, where like the bags that are our bodies are not designed to live or to function forever in an unchanging state. And as these different environmental and natural impacts affect our cells and our tissues, things change over time. So let me try to do a better job of explaining what molecularly we mean here, right? And I think that would actually help to maybe touch on the 9 hallmarks of aging really, really briefly, just so we know what we're talking about. At Cambrian, we separate these into 3 categories of changes that we think of as like changes inside the cell. So I already mentioned in cancer, we have mutations to the DNA, but also as our cells divide, the ends of our chromosomes called the telomeres, which you and some of the listeners may have heard of, they get shorter every time a cell divides, right? And so that's another kind of molecular damage in a way, because our cells are becoming less able to do more divisions every time they divide. So then there's also inside the cell a loss of what's called epigenetic information. So this is some of the markers on top of the DNA that change which genes are expressed in which cells at which time. And the more information is lost there, the less well the cells perform the job that they're supposed to do. Then we have a second category of like whole cellular level dysfunction. So cells themselves can become old and dysfunctional. We call this cellular senescence, where they were supposed to die, but actually fail to do so. And they start operating in this different program that spews inflammatory cytokines. So basically inflammatory molecules out into the environment. And they can cause quite a lot of havoc, even if you only have one out of every 100 cells being in this senescent state. And similarly, another kind of like whole cellular level dysfunction, we have the power plants in our cells, the mitochondria that make energy. Those start working less and less well over time as our bodies get older. And so the power plants can become dysfunctional, and there's a few different types of molecular damage that can make mitochondria not work well. So this is kind of a second theme of cellular level dysfunctions. And then the final theme is tissue level dysfunctions. So we have stem cells in our tissues, right? That are trying to regenerate, especially when we're young, regenerate injured organs. Our liver is super good at regenerating, for example. Our bone marrow, our immune systems are super good at regenerating. But as we get older, the stem cells that are fueling all of that, they get worn out over time. And they are able to regenerate less and less well. Another one that's in this kind of like organismal level, or whole tissue level, is the topic of chronic inflammation. So long-term chronic inflammation does horrible things to a tissue. It turns it from being very flexible and flexible and regenerative to what we call fibrotic. Basically, we become more and more like a scar as we get older, not just on our skin, but in all of our tissues. And so we're spending a lot of time thinking about this increased scarification that happens not just in our skin, but in our liver, in our kidneys, our hearts, our lungs as we get older, and what molecular tools we could use to reverse some of that, to make our tissues more flexible, more able to regenerate, instead of hardening up. So that gets into a little bit of scientific detail, but hopefully gives a sense of like the types of damage that we are thinking about in this geroscience field.
Yeah, and what's so interesting about this, I think that there's a sequence that people may or may not understand, which is that a lot of this molecular damage happens in what most would consider your healthy years. And then it actually shows up, 10, 20, 30 years later. And so maybe talk through a little bit of like that sequencing around when the damage happens, but then also like when it shows up and how that's not actually happening simultaneously in most cases.
Exactly. I think that's the key to understanding this whole field, is that when someone is diagnosed with a disease like Alzheimer's, let's say, that Alzheimer's diagnosis it's like not the start of the disease. It's one inflection point of a really long process that's been happening in that person. And so what this field of aging biology or geroscience is helping people think about is that if we can understand all of the damage that's building up 10 years, 20 years, 30 years before what we call a phenotypic effect, right? So before someone is actually losing their memory or unable to walk or unable to eat or something like that that can happen to a patient with Alzheimer's, or in the case of, cancer, before you see a big tumor that's metastasizing. If we can understand that damage in the 10, 20, 30 years leading up to it, measure that damage, and then use a biomedical intervention to either stop it or reverse it before we get a disease, then it's a much easier problem to solve than it is to try to react to a disease once it's already happened. And I actually have like a kind of nerdy scientific example that I like to use to think about this. So as a VC in bio, we invest in companies like cancer companies, for example, right? And one of the ways that drugs are tested in cancer companies is you give a mouse a tumor and then try your experimental treatment to see if the mouse can clear out its tumor, right? And so it's pretty standard practice. You're giving mice cancer, curing the cancer, and then you can use that as a justification for trying it in humans later. But as a VC, one thing that we all do is we make sure in these mouse models of cancer that before we try our experimental treatments, that the mouse has been injected with a tumor, and the tumor is grown big and scary enough that it's going to be hard for the treatment to clear out that tumor. Because if you treat the mouse too early, when the cancer is just developing, we know that almost every anti-cancer treatment works really, really well, and you get 100% cure rates. But that doesn't then translate into good results for patients, human patients that have advanced tumors. And so it's this fascinating situation to me, and I guess a bit nerdy here, that we're turning the thinking on its head, where a lot of the VC and investment thesis in how we think about mouse models has been, how do we make sure we can replicate advanced stage human disease in the mice so that the mice that we're treating look more like the patients that we're treating? That's one way to think about this investment thesis. But this geroscience hypothesis almost turns it on its head, which is if we know a way that we can cure cancer almost every single time, why don't we start thinking about how we treat our human patients in the way that works almost every time in our mouse models? And I think that kind of flipping of the script is one of the powerful ideas behind this.
One of the most fascinating things to me in all of this was when I learned about Brett Weinstein and kind of a lot of the controversy around the mice and the shortening of telomeres and kind of how a bunch of the mice came from the same place. And without getting super into detail about that whole situation, people can go Google it. But it really just showed that hey, this is still science. And if you don't get kind of a bunch of stuff right, the conclusion can be inaccurate. And so this whole idea of what's the framework you use, right? Do you try to replicate the human disease in a mouse and then go ahead and address it? Or do we just figure out how do we cure mice and then should we bring that to the humans is a fascinating way to kind of look at it. What I want to talk about next is this idea of healthy years. And Christian and I have talked a ton about this. And I think that a lot of people, when they think of longevity, they just simply say, okay, well, if the average human lifespan is 77, 78 years, whatever it is now, how do we get that to 80? And then how do we get that to 85, 90, 100? And that makes sense because you just want to live more years. But actually, there's a difference between living for an extra 20 years at the healthy or unhealthy state of a 90-year-old versus living an extra 20 healthy years that might actually be more what you feel and how you operate at 45, not actually feeling the effects of older age until maybe you're 65, and then being able to kind of capture those healthy years. So talk a little bit just how you think about healthy years versus maybe non-healthy years and why it's so important to focus on the former rather than the latter.
Yeah, I think you nailed our thesis right off the bat there, which is if we only extend the unhealthy period at the end of life, we're doing a disservice rather than a service to the population at large. So anything that's worth doing has to extend the number of healthy years. And that's our ultimate readout. So whenever we talk about it, we actually avoid the term lifespan altogether unless it has the term healthy put before it. Or I think the most popular term among people in our field is actually to talk about it in terms of health span, as one word, or my favorite, again, kind of wonky term, which is QALYs, the quality adjusted life years, where this is kind of how insurance companies think about, quantifying human life happiness, kind of that cross product. And so the positive news here is what we've seen both in humans as our lifespans have risen from 35 at 120 years ago at the start of the 1900s to close to 80 now. We've seen as human lifespan has gone up, human healthy lifespan has also gone up dramatically. Obviously, because we're dying of these chronic and horrible diseases like cancer and cardiovascular disease and Alzheimer's now, there is a tail period of unhealthiness that most of us experience at the end of our lives. But in humans, we have been able to extend that healthy lifespan portion out very significantly. And in mice and other model organisms that we've been able to use drug interventions to extend those the healthy lifespan of those animals, you're basically never able to increase just the unhealthy portion of life. Because one of the conclusions, or I guess the assumptions from this damage theory of aging, is that once you've got a critical amount of damage that's going to send you kind of tumbling downhill, which will ultimately result in the organism's death, like you can't undo that hill from that inflection point. So it's a much simpler idea to extend the time before you reach that turning point that starts you tumbling downhill. And so when I talked a little bit about like the mouse life extension earlier, you extend, like the way that essentially all of these treatments work is you extend that healthy period, and then the period of decline that ultimately ends in the death of the organism actually stays fairly constant, because once you've reached that big threshold, then there's almost just nothing you can do, and it's a matter of time.